Hello students, welcome to Zoology classroom. This is L. Mahesh, Assistant Professor of Zoology from Tara Government College, Sangareddy. In today's class, we are going to discuss about Porifera general characters. This topic comes under BSc first year Zoology subject in first semester. So Porifera is the second phylum in animal kingdom. This is the first multicellular organisms. It is containing a multicellular organisms. So even though they are multicellular organisms, the poriferans are multicellular organisms, but they are not, the cells of them are not developed into tissues. Hence, they are not considered as a real metazoans. So this Porifera has put in a separate group called as Parazoa. So the animal kingdom is classified into three subkingdoms. One is Protozoa, second one is Parazoa, in which this Porifera phylum fits in, and third one is Eumetazoa. In Eumetazoa, all the phyla that comes after this Porifera are placed. So Porifera is a phylum which consists of multicellular animals, but they are not real metazoans. Instead, they are placed in a separate group called as Parazoa, and this Parazoa remains as a blind stalk at the beginning of the phylogenetic tree of animal kingdom. And for the first time, a Scottish anatomist and zoologist, Robert Edmund Grant, proposed the name Porifera. The name Porifera originated from two words, two Latin words. They are porus and ferre. In Latin, porus means pores or holes, and ferre means tubule. As the organisms that come under this Porifera phylum are having many pores on their body surface. Hence, the phylum got this name, Porifera. So, Robert Edmund Grant has proposed Porifera name for these animals, for these organisms. And all the organisms that come under this Porifera phylum are known as sponges. And they are the simple type of aquatic invertebrates. In this phylum, so far 10,000 species of sponges have been identified. Among them, most of the species are marine organisms, while only some few, like 100 species only, are freshwater sponges. So most of them are living in marine habitats only. Only 100 species are identified from freshwater habitats. So for the first time, when scientists have seen these organisms, they regarded or they considered these organisms as plants. Why? Because they are not showing this motility or mobility. A feature that is seen in almost all animals except few. So as they are sedentary organisms and as they look like plants, they were considered as plants only. Later, the investigations, the revelations have led to the conclusion that poriferans are not plants actually, but they are also animals. And their body is asymmetrical or radially symmetrical, and with a variety of sizes and colors. So in terms of symmetry, many of the poriferans, they show two types of symmetry. One is asymmetry. That means they do not show any symmetry. Their body is irregular, so that we cannot get any symmetrical halves through any plane of those organisms' bodies. While some organisms can exhibit radial symmetry, so some are radially symmetrical and some are asymmetrical. And coming to the sizes also, varying sizes can be seen in these organisms. And in terms of colors also, uh, dull colors to vibrant colors. So a range of variety of colors are also observed in the poriferans bodies. And the colors are attributed to the presence of some other symbiotic zooxanthellae. So due to the presence of these chlorophytons only, the organisms, they will get different types of colors. And the habitat of these organisms, they can prefer to live in tidal zones to almost 9,000 meter depth ranges. And most of these sponges are attached to submerged hard rocks during their adult life. When they are spending a larval life, during that time only, for a short period of time, they swim freely in the water. And when they attach to the substratum, then they will start developing into adult ones. So during the adult life and in the life cycle, most of the life cycle, most time of the life cycle is spent in a sedentary life. And in this sedentary life, their bottoms are attached to the hard substances. Mostly those hard substances will be submerged hard rocks or any hard substance. And as we have already discussed, the poriferans are commonly called as sponges. And the shape of body is cylindrical and are vase-like. And some poriferans also available in round shapes. And body is radially symmetrical in few sponges, and in many sponges, they do not show any symmetry as their body is asymmetrical or their body is irregular in shape. And shapes also, variable irregular shapes are possible in these organisms. And sizes also, small size to a large size organisms are also observed in this file. And they are sedentary, aquatic animals, mostly marine, but a few are freshwater animals. So this is a typical example for the poriferan cycon. The other name for this organism is Skypha. So in this organism, we can see that the bodies are cylindrical or vase-like. And as these are attached to a single base, we can consider that this is a colonial organism. So as they are not symmetrical, they are irregular in shape at this bottom part, so they cannot be considered as a symmetrical thing. But when we consider each individual, we can get radial symmetry in the individuals. And as we have seen in last slide, some are colonial sponges and some are single organisms or solitary organisms. And they are multicellular. We have already discussed about this part, multicellular, but tissues are not developed in these organisms. And their body base two layers in their bodies. So they are considered as diploblastic animals. So in these two layers, the outer layer is called as pinacoderm, and the inner layer is called as choanoderm. And between these two layers of pinacoderm and choanoderm, a third layer also exists that is called as mesophyll or mesenchyme. So, and in this mesenchyme, various kinds of amoeboid shaped cells are present, and these amoeboid shaped cells are called as amoebocytes. And body surface of these organisms, of all these organisms bears numerous pores, and these numerous pores are called as ostia. Singular for this pore, ostia, is ostium. So through this ostia only, water enters into this body via canal system, and finally into the central cavity of their bodies. That central cavity of their bodies is called as sphingocele. So, and at the top of the body, a single large opening will be there. In some organisms case, multiple large openings also may be there, and these openings are called as osculum. So these are having a special system in their bodies that is called as canal system, and this canal system helps to take the water into their bodies. Through this water currents only, they will perform different types of functions like digestion, respiration, excretion, and reproduction also. And as they do not have nose, as they do not have mouth, there is no question of having digestive system and sense organs. So as we have already discussed, these are multicellular organisms, but they do not they do not have uh, tissues or organ systems. So there is no question of having any digestive system or any sense organs. So no tissues are present, no organs are present, no organ systems are present, only cells are present, and these cells are not developed into tissues. So intracellular digestion occurs in their body, and the nutrition type is holozoic. So, and they do not have any special excretory in the respiratory systems or respiratory organs. So the body surface and the inner surface of these canal systems helps in the excretion of waste materials and respiration process. So in the mesenchyme, there are different types of spicules are present, and these spicules are made up of calcium carbonate or silica. So when they are made up of calcium carbonate, they are called as calcareous spicules, and when they are made up of silica, they are considered as siliceous spicules. So some sponges may have, along with the spicules, are solely spongin fibers also. So the spongin fibers and spicules, they are making the one type of skeleton in their bodies. These spongin fibers and spicules, they will give support to the big size bodies. And they feed on organic particles and minute small organisms. And these food particles and minute organisms, they enter into the body of these organisms via the water currents, via this canal system. And all these food particles are ingested by the flagellated cells or choanocytes, which are lining the choanodome. So both type of asexual and sexual reproduction methods are possible in these organisms. Asexual reproduction occurs by forming external or internal budding. So as we have seen in the case of protozoans, so budding starts as a small protuberance which grows on the outside of the body, or it may also develop inside the body of the sponges also. If it is inside, it is called as internal budding. If it is developing on the outside of the body, then it will be called as external budding. So, and once the bud grows into a full size, once it develops the canal system and sphingocele and all required body parts, the fully grown bud may cleave off from the mother's body and may lead an independent life, or it may continue with the mother's body to form a colony. colony. So in this way, budding is one type of asexual reproduction. And another asexual reproduction is also possible, that is called as gemmules. So the gemmules are formed during the harsh conditions, or in other words, we can say unfavorable conditions. So during unfavorable conditions, the sponge will disintegrate. Before it disintegrates, before it dies, it will form a type of special spores-like structures called gemmules. So in these gemmules, some of the amoebocytes will be present. These amoebocytes are archaeocytes, I must say. So archaeocytes will be present. These archaeocytes, when again the favorable condition return, they will develop a new sponge. So, and one more thing, regeneration is also possible. Regeneration also may be regarded as one type of asexual reproduction method in these organisms. And the sponges, they exhibit a highest level of regeneration capacity in the whole animal kingdom. When the organism is cut into small tiny pieces, if a small tiny piece, if it consists one or few archaeocytes, each of those pieces, they will ultimately develop into many sponges. So in this way, the regeneration also leads to the one type of reproduction, asexual reproduction. And sexual reproduction is also present in these organisms. And the sexual reproduction is made possible with the fertilization of gametes. Fertilization is external, and development is indirect through two types of larvae, such as amphiblastula and ragonide larvae can be developed in these organisms. And there are no special gonadial organs, or gonads are not present, as we have already discussed. They are multicellular but lacking tissues and organs. So they do not have any gonads. So the cells which are having the capacity of this pluripotency, they are called as archaeocytes. These archaeocytes only, they will form the gametes, male gametes and female gametes. Male gametes and female gametes, both the gametes will be formed in the same individual, but self-fertilization is not possible. Why? Because a protandry or protogamy condition will be there. So both the gonads will not be formed at the same time in this same individual. So invariably, the organisms are forced to practice external fertilization. So in external fertilization, the sperms are male gonads, male gametes developed in the individual are released into the water through this water current, which enters into this body via canal system, and the water which leaves the body through osculum. These gametes, male gametes, are also sent outside of the body. So once the gametes are outside of the body and through the water currents, these gametes will enter into the bodies of another gamete, in the body of another sponges. In the body of those another sponges, they will meet with the female gametes, and thus fertilization takes place inside the body of the uh, organism. So in this way, fertilization is internal actually. In this slide, it is mentioned as external, but fertilization is internal in most cases. So, and in the development, larva will be there. So when there is a larval stage in the development process, in the development cycle, it is considered as indirect development. So development is indirect as larvae are there. And apart from these features, these cells are having different types of cells in them. So these different types of cells, they perform different types of functions in these in the organism's body, in the poriferans. And those functions are those cells are different types of cells are pinacocytes. They are considered as dermal cells or epidermal cells. They form the external surface of the body. And in the pinacoderm, here and there, some porocytes are there. And these porocytes are responsible for the forming of ostia or minute pores. So through this ostia only water enters into the body. And second type of cells are, third type of cells are choanoderms. They are also called as flagellated cells. These flagellated cells are responsible for drawing the water into the body as they are having a flagellae. The movement of flagellae creates water waves. These water waves bring the outside water into the body of these sponges. So choanocytes help in the forming water currents and drawing the water into the sponge body. So apart from this water current creation, drawing the water into the body, these choanocytes are also responsible for engulfing food particles. So they also help in the nutrition function, and in the sexual reproduction also. These choanocytes, they will engulf the external male gametes, and they will transmit this, or they will transfer this external or foreign male gamete to their self female gamete. So in this way, choanocytes are helpful in many ways. They create water currents, they bring water into the body, they help in nutrition function by engulfing food and creating food food vacuoles, and they also uh, help in respiration function. They also help in excretion function. They also help in sexual reproduction also. So this is a third type of cell, choanocyte. And apart from these three types of cells, multiple other cells are also present. Of them, important ones are archaeocytes. So archaeocytes are said to be pluripotent. They can form any type of cell whenever there is a necessity arises. So they can create and all other types of cells. So they have the regeneration capacity. So they are pluripotent cells. These pluripotent cells only are responsible for producing gametes. And thisocytes are also present. Thisocytes, they will store the food reserves in the body, and the chromocytes, they are responsible for they are responsible for the body colors of the sponges. And amoebocytes, these amoebocytes, they wander in the mesoglea or mesenchyme of the body. So by wandering in the body, they will transfer materials from here and there. And some other cells, sclerocytes, are also present. These sclerocytes, they are responsible for the production of spicules. So the sclerocytes are two types of sclerocytes are the silicoblasts and calcoblasts, and spongioblasts are also third kind of sclerocytes. Spongioblasts are also present. They will produce spongin fibers in the sponge body. So these are the different types of cells which help in the different functions in the sponge body. So this is about the general characters of Porifera. With this, today's session is over, and in next class we will meet with another topic. Till then, bye.